Hi, my name is Rachel McCoy, and welcome to a world full of bacteria. Here is one question that many people ask daily. What are bacteria? Bacteria are single-celled organisms, or prokaryotes, that contain many unique characteristics. Bacteria can be found in almost every environment on Earth. There are thousands of different species named by man. Bacteria can be either helpful or harmful, depending on several aspects. I will explain later in the presentation. Most bacteria cells contain a cell wall that is comprised of plasma membranes and peptidoglycan. Peptidoglycan is a molecule complex of polysaccharides linked by amino acids. Bacterial cells do not have a nucleus. Instead, they contain a nucleoid, an area that is not surrounded by a membrane and contains the genetic material, or DNA. Bacterial cells can also contain ribosomes and cytoplasm. Some bacteria, like the one shown in the diagram, contain a tail-like filament called a flagellum, which is used for movement. There are several ways bacteria are classified. One way is through gram staining. Gram-positive bacteria have a thicker peptidoglycan layer in the cell wall, causing it to stain a darker purple, whereas gram-negative bacteria stain a lighter purple or clear color. In gram-negative bacteria, the peptidoglycan layer is sandwiched between two plasma membranes. Another way to classify bacteria is through the cell's shape. There are three basic shapes of bacterial cells: spirili, bacilli, and cocci, which are shown in order in the pictures below. Spirili bacteria are spiral-shaped, such as a helix. Bacilli bacteria are rod-shaped, and cocci bacteria are shaped as spears. A bacterial cell's metabolism is also used to classify bacteria. For example, depending on a bacterial cell's need for oxygen, the cell is classified as either an obligate anaerobe, a facultative anaerobe, or an aerobe. An obligate anaerobe cannot function while oxygen is present. A facultative anaerobe can function with or without oxygen, and an aerobic bacteria requires oxygen to function. Photoautotrophic bacteria are photosynthetic, meaning that they convert solar energy into nutrients. Chemoautotrophic bacteria carry out chemosynthesis, a process in which the bacteria oxidize inorganic compounds to obtain energy needed to gain nutrients. Finally, chemoheterotrophic bacteria take in organic nutrients through absorption. Another question that many people ask: are bacteria helpful or harmful? To answer that question, it depends on the species of bacteria. There are several species of bacteria that are helpful to humans, such as E. coli. E. coli is found in the human digestive tract and aids in digestion. There are also several bacteria species that cause illnesses or diseases in humans. For example, that bacteria Streptococcus causes strep throat and Staphylococcus causes MRSA or staph. Harmful bacteria can cause infections in the human body and can lead to death in severe cases. One way to combat harmful bacteria is through the use of antibiotics. Antibiotics are drugs prescribed by a doctor that rid the body of bacteria by inhibiting either the protein biosynthesis or the cell wall biosynthesis. However, because of the heavy usage of antibiotics, bacteria have begun to mutate or adapt to become resistant to antibiotics. There are some bacterial cells that are resistant to every antibiotic known to man, creating several serious problems for the person infected. There are several bacteria that are helpful in regard to the environment. Some bacteria are decomposers that break down organic materials in the soil, creating usable nutrients for other living organisms. Some bacteria cycles nutrients and transforms them into usable forms. For example, bacteria can take in carbon and change it to carbon dioxide, which is necessary for plants to undergo photosynthesis. Another way bacteria is helpful to the environment is the transformation of nitrogen into nitrates or nitrites, which are used by plants to grow. And now, to my final thoughts on bacteria. There are still many bacterial species being discovered by scientists today, and advancements in science and technology regarding bacteria are still being made. Although we understand a lot about bacteria, there is never a limit to our knowledge. We still have the issue of bacterial resistance to antibiotics, as well as finding other ways to combat harmful bacteria. In the future, my goal is to play a role in finding a solution to this problem and hopefully help save thousands of lives that are stolen by diseases caused by bacteria.